Chapter 14 Respiration in Plants. All of us breathe to live, but why is breathing so essential to life? What happens when we breathe? Also, do all living organisms, including plants and microbes, breathe? If so, how? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know we eat food for energy, but how we know we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms for their energy requirements, do they eat food? You may have wonder you may wonder at the several questions raised above. They may seem to be very disconnected, but in reality the process of breathing is very much connected to the process of release of energy from food. Let us try and understand how this happens. All the energy required for life processes is obtained by oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food by the process of photosynthesis. They trap light energy and convert it into chemical energy that is stored in the bonds of carbohydrates like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissues, and organs photosynthesis photosynthesize. Only cells containing chloroplast that are most often located in the superficial layers carry out photosynthesis. Hence, even in green plants, all other organs, tissues, and cells that are non-green need food for oxidation. Hence, food has to be translocated to all non-green plants. Animals are heterotrophic, that is, they obtain food from plants directly. Herbivores or indirectly carnivores. Saprophytes like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life processes comes from photosynthesis. This chapter deals with the cellular respiration or the mechanism of breakdown of food materials within the cell to release energy and the trapping of this energy for synthesis of ATP. Photosynthesis, of course, takes place within the chloroplast in the eukaryotes, whereas the breakdown of complex molecules to yield energy take place in the cytoplasm and in the mitochondria, also only in eukaryotes. The breakdown of the carbon-carbon bonds of the complex compounds through oxidation within the cells leading to release of considerable amount of energy is called respiration. The compounds that oxidize during this process are known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substances in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrates is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be used directly, but is used to synthesize ATP, which is broken down whenever and wherever energy needs to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in the ATP is utilized in various energy requiring processes of the organisms, and the carbon skeleton produced during the respiration is used as precursors for biosynthesis of other molecules in the cell. 14.1. Do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require O2 for respiration to occur, and they also give out CO2. Hence, plants have systems in place that ensure the availability of O2. Plants, unlike animals, have no specialized organs for gaseous exchange, but they have stomata and lenticels for this purpose. There are several reasons why plants can get along without respiratory organs. First, takes plant each plant part takes care of its own gas exchange needs. There is very little transport of gases from one plant part to another. Second, plants do not present great demands for a gas exchange. Roots, stems, and leaves respire at rates far lower than animals do. Only during photosynthesis are very large volumes of gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. When cells photosynthesize, availability of O2 is not a problem. In these cells, since O2 is released within the cell, third, the distance that gases must diffuse even in large bulky plants is not great. Each living cell in a plant is located quite close to the surface of the plant. This is a true This is true for leaves. You may ask, but what about thick, woody stems and roots? In stems, the living cells are organized in thin layers inside and beneath the bark. They also have openings called lenticels. The cells in the interior are dead and provide only mechanical support. Thus, most cells of a plant have at least a part of their surface in contact with air. This is also facilitated by the loose packing of parenchyma cells in the leaves, stems, and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which provides the CO2 and H2O as end products, yields energy, most of which is given out as heat. C6H12O6 plus 6O2 gives a 6CO2 plus a 6H2O plus energy. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules than the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecule in such a way that not all liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy release can be coupled to ATP synthesis. How this is done is essentially the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide, water, and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of such situations and organisms where O2 is not available? There are sufficient reasons to believe that the first cells on this planet lived in an atmosphere that lacked oxygen. Even among present-day living organisms, we know of several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in others, the requirement for anaerobic conditions is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. 14.2 Glycolysis. The term glycolysis has originated from the Greek words glycos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Emden, Otto Meyerhoff, and J. Parnas, and is often referred to as the EMP pathway. In anaerobic organisms, this is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis or for storage. Carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of the enzyme hexokinase. Thus, phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure 14.1. In glycolysis, a chain of 10 reactions under the control of different enzymes takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please take note the steps at which the utilization of synthesis of ATP, or in this case NADH plus H plus, takes place. ATP is utilized at two steps: first in the conversion of glucose into glucose 6-phosphate, and second in the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. The fructose 1,6 bisphosphate is split into hydroxyacetone phosphate and 3-phosphoglyceraldehyde PGAL. We find that there is one step where NADH plus H plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde PGAL is converted to 1,3 bisphosphoglycerate BPGA. Two redox equivalents are removed in the form of two hydrogen atoms from PGAL and transferred to a molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid PGA is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP synthesized during the conversion of PEP to pyruvic acid. Can we then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Pyruvic acid is then a key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic conditions in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt a Krebs cycle, which is also called as anaerobic respiration. This requires O2 supply. Figure 14.1 steps of glycolysis. Glucose, which is a 6 carbon, gives glucose 6 phosphate, which is 6 carbon, ATP to ADP, which gives fructose 6 phosphate, which is also 6 carbon, ATP to ADP, which gives fructose 1,6 bisphosphate, 6 carbon, which gives triose phosphate, glyceraldehyde 3 phosphate. Which isomerizes to triose phosphate dihydroxyacetone phosphate, which is also 3 carbon, NAD plus 2 NADH plus H plus, gives 2 times of triose of bisphosphate, 1,3 glyceric acid, 3 carbon, ADP to ATP gives 2 times of triose phosphate, 3 phosphoglyceric acid, which is also a 3 carbon compound, which gives 2 times of 2 phosphoglycerate, which gives 2 times of phosphoenol pyruvate in presence of H2O, and which gives 2 pyruvic acid, which is a 3 carbon compound from ADP to ATP in conversion of ADP to ATP. 14.3 Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic conditions by sets of reactions where pyruvic acid is converted to CO2 and ethanol. Enzymes, pyruvic acid decarboxylases, and alcohol dehydrogenase catalyze these reactions. Other organisms, like some bacteria, produce lactic acid from pyruvic acid. The steps involved are shown in figure 14.2, and animal cells, like muscles, during exercise when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the processes. In both lactic acid and alcoholic fermentation, not much energy is released, less than 7% of the energy is in glucose is released, and not all of it is trapped as high energy forms of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is the net ATPs that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis. When one molecule of glucose is fermented to alcohol or lactic acid, yeast poison themselves to death when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think the alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecules needed for cellular metabolism? In eukaryotes, these steps take place within the mitochondria, and this requires O2. Aerobic respiration is the process that leads to a complete oxidation of organic substances in the presence of oxygen and releases CO2, water, and a large amount of energy present in the substrate. This type of respiration is the most common in higher organisms. We will look at these processes in the next section. Figure 14.2 Major pathways of anaerobic respiration. Glucose gives glyceraldehyde 3 phosphate, NAD plus 2 NADH plus H plus 3 phosphoglyceric acid, which gives phosphoenol pyruvate acid, which gives pyruvic acid. And from that it gives ethanol plus CO2, NADH plus H plus 2 NAD plus, and from pyruvic acid it gives lactic acid, that is NADH plus H plus 2 NAD plus. 14.4 Aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, is transported from the cytoplasm into the mitochondria. The crucial events in aerobic respiration are: first, a complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of CO2. Second, the passing on of the electrons removed as a part of the hydrogen atoms to molecular O2 with the simultaneous synthesis of ATP. What is interesting to note is that the first process takes place in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of the carbohydrates in the cytosol after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions characterized by pyruvic dehydrogenase. The reaction is catalyzed by pyruvate dehydrogenase require the participation of several coenzymes including NAD plus and coenzyme A, pyruvic acid plus OA plus NAD plus in the presence of Mg2 plus ions, and pyruvate dehydrogenase gives acetyl CoA plus CO2 plus NADH plus H plus. During this process, two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid produced from one glucose molecule during glycolysis. The acetyl CoA then enters the cyclic pathway 5-carboxylic acetyl cycle, more commonly called as the Krebs cycle, after the scientist Hans Krebs, which first elucidated it. 14.4.1 Tricarboxylic acid cycle. The ECA cycle starts with the condensation of the acetyl group with oxaloacetic acid OAA and water to yield citric acid. 14.3 A reaction is catalyzed by the enzyme citrate synthase and a molecule of CoA is released. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation leading to the formation of alpha-ketoglutarate acid and then the succinyl-CoA. In the remaining steps of citric acid cycle, succinyl-CoA is oxidized to OAA, allowing the cycle to continue. During the conversion of succinyl-CoA to succinic acid, a molecule of GDP is synthesized. This is a substrate-level phosphorylation. In a coupled reaction, GDP is converted to GDP, which is a simultaneous reaction of ATP from ADP. Also, there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus, and 1 point where FAD plus is reduced to FADH2. The continued oxidation of acetyl CoA via the TCA cycle requires the continued replenishment of oxaloacetic acid. The first member of the cycle, in addition, it also requires the regeneration of NAD plus and FAD plus and from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows: pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ADP plus IP gives, in the presence of mitochondrial matrix, gives 3CO2 plus 4 NADH plus 4H plus FADH2 plus ATP. We have till now seen that the glucose has been broken down to release CO2 and 8 molecules of NADH plus H plus 2 of FADH2 have been synthesized besides just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither O2 has come into the picture nor the promised large number of ATP has yet been synthesized. Also, what is the role of NADH plus H plus and FADH2 which is synthesized? Let us now understand the role of O2 in respiration and how ATP is synthesized. Figure 14.3 The citric acid cycle. Pyruvate 3-carbon coenzyme A NAD plus gives NADH plus H plus CO2 release acetyl coenzyme A 2-carbon chain which gives citric acid with 6-carbon CO2 is released. NAD plus gives NADH plus H plus alpha ketoglutarate acid 5-carbon CO2 release. NAD plus gives NADH plus H plus GDP to GTP. Succinic acid, which is a 4-carbon compound. FAD plus to FADH2. Malic acid, which is a 4-carbon compound. NAD plus gives NADH plus H plus oxaloacetic acid, which is 4-carbon compound, and then the cycle continues. 14.4.2 Electron transport system, ETS and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to the O2, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another is called electron transport system, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during the citric acid cycle are oxidized by NADH dehydrogenase complex 1, and electrons are then transferred to ubiquinol located within the inner membrane. Ubiquinol also receives reducing equivalence by FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electrons to cytochrome C via the cytochrome BC1 complex, the complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between complex 3rd and 4th. Complex 4th refers to the cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper centers. When the electrons pass from one carrier to another via complex first to fourth in the electron transport chain, they are coupled to ATP synthase complex fifth for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to three molecules of ATP, while that of one molecule of FADH2 gives two molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the process of oxygen is vital, since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor, unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation. In respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of the membrane liquid ATP synthesis as explained by chemiosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase complex 5, complex 5th. This complex consists of two major components, F1 and F0. The F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which the protein crosses the membrane. The passage of proteins through the channel is coupled to the catalytic side of the F1 component for the production of ATP. For each ATP produced, the 2H plus passes through F0 from the intermembrane space to the matrix down the electrochemical proton gradient. Figure 14.4 Electron Transport System (ETS). Intermembrane space, inner mitochondrial membrane matrix. NADH plus H plus gives NADP plus NAD plus complex 1 NADH dehydrogenase 2 electrons FMN FES complex 4H plus released out to intermembrane space electrons UQ, that is ubiquinol UQH2 ubiquinol electron complex third cytochrome BC1 cytochrome B FES complex cytochrome C1 4H plus ends out cytochrome C intermembrane space cytochrome C complex 2 succinate dehydrogenase succinate to fumarate FAD plus FAD to FES complex to UQ to UQH2 and electrons back to cytochrome B. And from the intermembrane space, cytochrome C is Cu, cytochrome A, then cytochrome A3, then Cu. Half O2 plus 2H plus ends out. Intermembrane space, complex 4, that is the cytochrome C oxidase. F1 complex, F1 ATP synthase, ADP plus IP is ATP, H plus ends out to the matrix, left for chemical gradients. (14.5) Diagrammatic representation of ATP synthesis in mitochondria. Inner mitochondrial membrane, outer side matrix, F0 and F1, ADP, AP to ATP, 2H plus ions. 14.5 The respiratory balance sheet. It is possible to make calculation of the net gain of ATP for every glucose molecule oxidized, but in reality this can remain only a theoretical exercise. These calculations can be made only on certain assumptions that first, there is a sequential orderly pathway functioning with one substrate following the next, and with the glycolysis TCA cycle and ATS pathway following one after another. Second, in ADPH synthesizing glycolysis is transferred into The mitochondria undergoes oxidative phosphorylation. Third, none of the intermediates in the pathway are utilized to synthesize any other compound. Fourth, only glucose is being respired. No other alternative substrates are entering the pathway at any of the intermediary stages, but this kind of assumptions are not really valid in the living system. All pathways work simultaneously and do not take place one after the other. Substrates enter the pathways and are withdrawn from it as when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means, yet it is useful to do the exercises to appreciate the beauty and efficiency of the living systems in extraction and storing energy. Hence, there can be a net gain of 38 ATP molecules during the aerobic respiration of one molecule of glucose. Now let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to CO2 and H2. In fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions. And the pH is oxidized to Nd plus atom slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. 14.6 Amphibolic pathway. Glucose is favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrates can also be respired, as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step. See figure 14.6 to see the points of entry of different substrates in the respiratory pathway. Fats would need to be broken down into glycerol and fatty acids first. If fatty acids were to be respired, they would first be degraded to acetyl-CoA and enter the pathway. Like glycerol would enter the pathway after being converted to TGAL. The proteins would be degraded by proteases and individual amino acids after deamination, depending on the structure, would enter the pathway at some stage within the Krebs cycle or even as pyruvate or acetyl-CoA. Since respiration involves breakdown of substrates, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. But is this understanding correct? We have discussed about at which points in the respiratory pathway different substrates would enter if they were to be respired and used to have energy. What is important to recognize is that it is that it is these very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrates. Hence, fatty acids would be broken down to acetyl-CoA before entering the respiratory pathway when it is used as a substrate. But when the organism needs to synthesize fatty acids, acetyl-CoA would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into the picture both during the breakdown and synthesis of fatty acids. Similarly, during the breakdown and synthesis of protein 2, respiratory intermediates from the link breaking down processes within the living organisms is catabolism and synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one. 14.6 Interrelationship among metabolic pathways showing respiration mediated breakdown of different organic molecules to CO2 and H2O. Fats, carbohydrates, proteins, fatty acids, and glycerol. Acetyl-CoA would cycle H2O and CO2 out. And then glycerol dehydroxyacetone phosphate will cell process glycerol dehyde 3-phosphate, which gives pyruvic acid, which gives acetyl-CoA, which gives the Krebs cycle. Carbohydrates, simple sugars, that is, for example, glucose, which gives glucose 6-phosphate, which gives fructose 1,6 bisphosphate, which gives dihydroxyacetone phosphate reversal process glycerol D3-phosphate, which gives pyruvic acid, which gives acetyl-CoA, which gives Krebs cycle. Proteins gives amino acids, which gives pyruvic acid, and acetyl-CoA and Krebs cycle. 14.7 Respiratory quotient. Let us now look at another aspect of respiration. As you know, during aerobic respiration, O2 is consumed and CO2 is released. The ratio of the volume of CO2 evolved to the volume of O2 consumed. And respiration is called the respiratory quotient RQ or respiratory ratio RQ. Volume of CO2 evolved upon volume of O2 consumed. The respiratory quotient depends upon the type of respiratory substrate used during respiration. When carbohydrates are used as substrate and are completely oxidized, RQ will be 1. because equal amounts of CO2 and O2 are evolved and consumed respectively as shown in the equation below. C6H12O6 plus 6O2 gives a 6 CO2 plus a 6H2O plus energy. Respiratory quotient 6 CO2 by 6O2 gives 1.0, that is 1. When fats are used in respiration, the RQ is less than 1. Calculations for fatty acid tripalmethyl, if used as a substrate, is shown 2 times of C51H98O6 tripalmethane plus 145O2 gives 102 CO2 plus 98 H2O plus energy. RQ is equal to 102 CO2 upon 145 O2, which is 0.7. When proteins are respiratory substrates, the ratio would be about 0.9. What is important to recognize is that in living organisms, respiratory substrates are often more than one. Pure proteins or fats are never used as respiratory substrates. Summary: plants, unlike animals, have no special systems for breathing or gaseous exchange. Stomata and lenticels allow gaseous exchange by diffusion. Almost all living cells in the plant have their surfaces exposed to air. The breakdown of carbon-carbon bonds of complex organic molecules oxidation cells leads to the release of a lot of energy. That's called cellular respiration. Glucose is the favored substrate for respiration. Fats and proteins can also be broken down to yield energy. The initial stage of cellular respiration takes place in the cytoplasm. Each glucose molecule is broken through a series of enzyme catalyzed reaction into the two molecules of pyruvic acid. This process is called glycolysis. The fate of the pyruvate depends on the availability of oxygen in the organisms. Under anaerobic conditions, either lactic acid fermentation or alcohol fermentation occurs. Unicellular eukaryotes and in germinating seeds. In eukaryotic organisms, aerobic respiration occurs in the presence of oxygen. Pyruvic acid is transported into the mitochondria where it is converted into acetyl-CoA with the release of CO2. Acetyl-CoA then enters the tricarboxylic acid pathway or Krebs cycle operating in the matrix of the mitochondria. NADH plus H plus and FADH2 are generated in the Krebs cycle. The energy in these molecules as well as that in the NADH plus H plus synthesized during glycolysis are used to synthesize ATP. This is accomplished through a system of electron carriers called electron transport system ETS located on the inner membrane of the mitochondria. The electrons as they move through the system release enough energy that are trapped to synthesize ATP. This is called oxidative phosphorylation. In this process, O2 is the ultimate acceptor of the electrons and it gets reduced to water. The respiratory pathway is an amphibolic pathway as it involves both anabolism and catabolism. The respiratory quotient depends upon the type of respiratory substance used during respiration.